Okay, so raise your hand if you were not able to open this file. Okay, you're still having issues. Did your trash delete? Okay, so you're redoing it. Okay, good. Anybody else not able to open these files? Cool. Okay, so again, what it is we're looking at is overall quality of how the run went. There's a lot of different things on here. You guys can learn more about them as well with Googling if you're very interested. I'm going to point out some of the most commonly used ones that I will actually look at. Honestly, a lot of these things like always fail and I just ignore it for whatever reason. But anyway, so per tile sequence quality, so what this is looking at is it's tiling the flow cell across the different regions, basically the window of the camera, and it's going to essentially show you if there was any particular regions that had a lower quality, maybe batch effects based on locations of tile and things like that. Honestly, this is something I never look at. Per sequence quality scores, this is one thing that I do look at. So the first graph was showing you the average quality across the base. This is showing you the average quality across the entire read. So it's really important that you have a full read with really good quality. And what you can see is that the average quality score is plotted down here. So our cutoff of 30, as in we're very confident, starts right here and goes forward this way. You can see that the vast majority of our sequences have a very high average quality across the entire read. So that's nice to see. That's something that I also take a look at. Per base sequence content, basically you would expect to have approximately random distribution of A's, T's, C's, and G's across your entire read. So that's what it's showing you here, the averages across these regions. You'll notice that the first one is on a single scale and then it kind of dips into like, I guess pairs of 2. So it averages out a little bit better. And the beginning is always a little bit funny with maybe adapters that were left over or random sequences like that. Yeah?
I don't know if you already answered this, but what did the check marks and the explanation points and the red X?
Okay, yeah, so these are recommendations that the program will throw out. The check mark means, based on the graph that we've made on your data, we think it looks good. Check, great. The exclamation mark is, it might be a little bit weird, maybe you should go in and look at this a little bit further. And the X means that it failed. It failed their standards according to the people that wrote this program and made these plots. So I don't pay too much attention to these. Again, each one tells you something very different. And what they call it as failing, you know, this still looks pretty good to me because across the vast majority of the read, I have a good average sequence quality. So you can look into it further and say if it works. If you're really curious to know more detail about any of these, you know, you can Google search exactly what it is that all of these are showing you on the graph. I don't really want to spend the time in class to go in depth into each one of them, but you can learn more about them and why maybe you see something that looks a little bit weird or why it might be potentially throwing a flag. Yeah? No. No. So you will upload your data, or yeah, go ahead.
So they will ask for your raw FastQ, so they can run FastQC themselves.
Right.
So if you want to download someone else's data from a paper that you see published, you can run FastQC and you should get exactly the same results. So you're not required to post these files because they're easily generated.
Right. So you upload your FastQs when you submit a paper.
Would you ever upload before that, and the raw file? And you absolutely have to put the raw data.
Yeah.
How much of the process data you put up, and depend on reviewers and the journal and
Yeah. And your field, you know, if you want to share your process data with the field and make it really easy, like the Encode website, for example, uploads a lot of process data as well. But it's also more work on the user, so depending on what your goals are, you know, and what they require, you can upload more or less of this stuff.
A lot of that is a philosophical discussion about open source code versus non-open source code and how much do you share and not share. Some PIs are really into open source, and if you write software or however you do, like some people will publish with their raw data their IPython notebook for exactly how they process everything, how every figure is generated, which is nice if you want to go in and reproduce exactly what you did. Not every PI is
Per sequence GC content, so again, you would expect that the average GC content of your read should be around 50%. It kind of shows your distribution and what the theoretical is. That looks fine. The length distribution is another one that I take a look at. We sequenced this 100 base pairs, so it's nice to see that all of our reads came out at 100 base pairs and nothing failed at the beginning or the end of the run that would be altering the length of our read. Okay. So I'm not going to talk about any more of this now. If you have more questions about it, we can talk about it later or after class, or you can always Google what each of these figures is showing you in more detail if you want to work on that. But I want to go ahead and move on to the next step. One more question? Yep.
When the adapters are turned off.
Yes, so Illumina does that for you when they give you the sequence. So remember in the FASTQ file we saw the sequence that was the index read. So what they give you off the machine is the clean trimmed sequences unique to that index. And again, they'll trim the index off for you. So all of the sequences that were associated with that index, only the insert that's your DNA piece will be included in that file for you. But they also, if they were not able to determine what the index was, they provide all of the files and then you can, decide where you want to go with them. But the FASTQ files that we're looking at are already trimmed in just the reads that were associated with a given index.
So some reason when that happens is sequencing errors. Another unfortunately common reason is when you submit your samples for sequencing, you fill out a manifest that says what the 6 base index sequence is, and it is not impossible to mess that up. And it's a huge pain for IBM or other students to go back and process the data to fix that. So just be careful.
Okay, so we've checked the general quality of the data sets that we're going to be using. They look okay, the run seemed to go okay. So we're going to go ahead and move on to the next step. If you go to 4, so this is the end of this notebook. We're going to talk about using annotations, where we get these annotations, how we use the annotations in the context of the specific aligner that we're going to use, and where all that comes from. So this next notebook, we're going to start using STAR. So we have our FASTQ file, we have our reads, and now we want to perform the alignment to the genome. Okay, so before we go into the alignment, I want to take a step back and comment on something specific about TSCC for when we're using these high processing jobs. So far, everything that we've done on TSCC, we've done on the command line. And we're using the processing of what's called the head node or the login node. So whenever you log in on your command line, you're given a designated amount of space. It's a very small amount of processing power, which basically gives you enough to do all the functions like moving around in directories, looking at files, transferring files around, doing very small things like FastQC, which we just did, had enough memory to be able to run on the login node. But a lot of the processing that we need to do, we need to reserve space. And what I was telling you guys about yesterday, TSCC is great. It's a cluster of giant computers in a room, and we often need to log into those computers specifically to handle the higher processing jobs. So we're going to do that by submitting jobs to the cluster. So this is different than simply logging into your head node, because rather than just using the small amount of space that everybody is given when they log in, we're going to reserve more space. And this is more efficient because you can imagine every time you log into TSCC, you don't need to reserve a lot of this space in all these computers. You only want to reserve it when you're going to do a high processing job. So we're going to reserve it specifically for the job using a submission script to specify how much space you need, how much time you need to run the job, and what job it is that you're going to be running. Okay, and then the submitted job goes into a designated queue. So I also mentioned this yesterday. TSCC is this room full of computers. Some labs have purchased space on TSCC. So as a Yo lab, we have designated computers that are reserved for us because we do this a lot, and we don't want to have to wait in the queue for this to happen. For other users, there's just general spaces where you can ask to reserve from. And we'll get into the different ones that we're going to be using in the class. So this is an example of what a submission script is going to look like into TSCC. It's called, actually I don't remember what it's called, but we use these PBS flags to describe the parameters of our job submission. So instead of now writing our commands on the command line in the head login node like what we've been doing so far, we're going to write scripts. And we're going to write scripts using a VI editor to make a file which includes all of the information about how we're going to be running something. So rather than running that line, your command on the line of your initial login, we're going to put that command in a script, which we're going to save in a file, and then we're going to submit that script as a command, and we're going to tell it, hey, everything that's in this script, we want you to do all of this stuff. So what you have to do when you're ready to submit a script is specify all of these submission parameters. So the first line at the very top specifies the language of the submission script that you're going to be using. So in this class we're using the bash programming. So this is going to say hashtag exclamation mark in bash. And this is an example of C shell. So we're going to change that to bin bash. And then each of these flags describe a different parameter of your job submission. So -q is what I was referring to as the queue name. You'll see the TSCC structure, and I'll show you which queues you have access to be able to submit to. This just basically means which reservation system you're going to put your job into and where it's going to be run and how it's going to be run. You can give your job a name so that you can track it. So this will be unique to each script that you run. You'll give it a very user-friendly name so that when it runs, it makes sense to you as to what it was that you were running. So if you were generating a star index, which is the first thing that we're going to do, we're going to give it the job name of generate star index. You guys should, again, this is just good computational practice to keep yourself organized, using things that make sense based on what you're doing. This line here is very important. Refers to the number of nodes and processors per nodes that you're reserving. So this is where you specify the amount of space and memory that you're going to be using for your particular job. So this is something that I'm going to be giving you for most of the jobs that we're running based on how much space I know these things require. If you're doing this on your own later on, you can always try things, and if it tells you like memory error, then you know you need to add more space, or sometimes the manual will give you guys useful suggestions on about where to start with this. So the way that this works is you're reserving nodes, and PPN stands for processors per node. So you can think of each computer as a given node, and then within each computer there's a bunch of processors that are running. So each node has a maximum of 16 processors per node. So when we are doing these reservations, it's best if you don't split your nodes, or you don't split your processors across multiple nodes, but you can reserve within one node up to 16 processors. And so that's what we're going to be specifying here. And again, this is something that I'll give you based on how memory intensive the job is that we're going to be running. So the first thing that we're going to do is going to require a lot of memory. So we're going to reserve all 16 processors on one node, and that's specifying into the queue. It's saying, okay, I need this job to run on 16 processors. So don't start my job until 16 processors are available for me to use. So that's what all of this is being specified for. Yeah.
How do you make decisions on how much memory you're using?
Yeah, so that's a really good question, and something that I honestly got because other people in my lab were running these programs, and I was like, hey, how much did you use to do this? And then it worked out well for me. So that's kind of the cheater's way to do it. You can also just try things. So if you try reserving a given amount based on what you know about the program and what it's trying to do, you can learn a little bit about, you can imagine that an alignment, okay, that kind of has a lot of stuff going on, that's probably going to need a lot, so you can try it, and if your job fails because of a memory error, then you know that you need to increase it. You can also refer to the manual, and they may or may not have helpful instructions for you there about how much memory is required to run it. Yeah?
Is there a difference, to having 10 nodes and only one queue processor per node?
Yeah.
Or like one node versus
Yeah, so this is something that I'm not super familiar with. I guess I didn't mention this on day one. My background is as a biologist, not a computer scientist. So I've been learning all of these things with less familiarity with how all of these systems are running and like the infrastructure behind it, and more from the biology side. So I'm sharing with you guys the things that I've learned from my lab mates, but there may be more completeness for this. Basically what I've learned is that when you have multiple nodes, you're splitting your job across different computers. And so that is oftentimes inefficient, and depending on the program that you're running, It may not even actually be using those other computers that you requested because the code will not actually let you split it across multiple computers. So it's always beneficial to reserve as many processors as you can on one individual node. So I always recommend keeping up to 16 processors on one node before actually, and when you go to more than one node, it's called splitting it across nodes, because there's some code out there that will work, you have to specify the program is actually able to do that, and depending on who wrote the code and how it was written, you may or may not be able to do that. So there could be a time where maybe you reserve 4 processors, or you reserve 4 nodes and 4 processors per node, and you submit your job, but your job is actually only running on one of those nodes because it can't be split, but all those other 3 are still reserved with your job, and you're just wasting space. So it's a good idea to try to reserve as many processors as you can on one individual node.
Will it always be 16 or does it depend on?
Depends on your job. So the maximum that you can hit is 16 processors per node. And we will use a couple different programs that have different amounts of memory required. And you'll see examples where we use all 16 processors on one node, and also examples where we request less than that. And this affects how long your job will sit in a queue. So when you're specifying these things, you're not able to actually run your job until this is available on TSCC. And depending on how many people are asking for stuff at the same time and how much you're asking for, your job will sit in the queue longer or shorter. So for example, if you have to wait for an entire 16 processors to free up before your job can be submitted, you might be sitting in the queue for a little bit longer than somebody who only needs 1 or 2 to free up, because as soon as those 1 or 2 free up, their job's going to get accepted and it's going to start running. Yeah.
Is RAM correctly timed the number of nodes?
That is beyond your scope of knowledge. It's just a reference of memory is how I understand it. And yeah, I'm not exactly sure how it's tied back to RAM. You can probably Google more about that. Okay, so in addition to specifying the amount of memory that you're going to use, you're going to specify a wall time, and this is going to say how long you think your job is going to be running. So this is where you're going to specify how long you want to make this reservation for. And there's limits on this, so because this is a shared cluster, there's certain queues that, because we're sharing this with a bunch of users, we don't want one user to request a job for 24 hours and jam up the queue for everybody else. So if you have longer processing jobs, you have to have your own space designated for it. So there's maximum of 8-hour wall time on the jobs that we're going to be running on a hotel queue. But the nice thing about wall time is once your job is actually done, your wall time will stop and it will time out, and it will cancel your reservation when your job is done. But the wall time that you provide, it should be an estimate, and it should always be a little bit of an overestimate, because if your job is still running but it's exceeded the wall, it's been running for longer than the wall time you specified, your job will die and it will say, okay, you've exceeded your wall time limit and your process is done. So this is something that you again will specify based on approximately how long you think your job is going to take. Yep.
So is this universal for all supercomputers that you can log into, or is this specific for TSCC?
So this is a particular type of submission script called PBS scripting, I believe. So depending on how the infrastructure of the supercomputer is set up, it may or may not use parameters that look like this. This is one common language, so this is not just TSCC, but a lot of clusters use this setup where you use PBS tags to describe the parameters of your submission. So it depends on the specific cluster. It's not 100% universal, but it's common, so it's used across other systems as well.
And the other TSCC website has a lot of this information.
Did you have another question?
Oh, are there other supercomputers that people use here, or is TSCC?
Not that I know of on campus. So TSCC is actually stored on campus. It's one that everybody uses here. I'm not sure everyone knows on campus. Okay. The other things are user specified. So -O is an output file. Again, this is very specific to your job. Name it something nice so that when you get an output file and it goes somewhere, you actually know what it means. So don't give it job number 1, but give it star generate index. Again, an error file. So any errors that are associated with your job are going to get put into this file. So the difference between this and the command that we've been doing on the head node so far, all of the outputs that you guys see on your head node pop up on your screen. But because we're writing a submission script and we're sending this away remotely to TSCC, it's only going to send the output back. So all of the intermediate stuff is going to get dumped in these output files and these error files. And you're not going to get to see those until the job is done and it's sent back to you. So it's really important that you specify the location, or you specify these files with a particular name so that when it comes back, it will make sense to you and you'll be able to find them and you'll know what they mean. Okay, we're not going to talk about the rest of these commands, but you can find more information about that. I provided the link somewhere on the TSCC website. So I'm just going to highlight the main ones that I use. So this is an example. Yeah.
Is there a way such that you can run the script and have an input on the command line that will be inputted into the script? So like say you want the output file to be something that you can designate on the command line and actually run the script. Is there a way to essentially like designate that as a variable that you load it into the script?
So when you specify submission parameters below all of this, you have commands, and in these commands you can have more than one. So if you want like the output file of your initial command to go as the input file into your next command, and you want to do a bunch of these things all together, you can specify that below, and we'll see a couple examples of that. I think that's what you're getting at?
I guess I'm more considered this like say I want my output file to go to a certain directory.
Oh yeah.
But I don't want to have to create a whole new script for that. Is there a way I can create a generalized script that on the command line when I say run this script and put it in this directory, it will run the script and then use that variable that I gave it to say this is the directory I want it to be sent to?
So that will all be specified in the specific command that you run. So just like when we ran fastqc, we specified the output directory of where we wanted those output files to go. So as you run a command down here, if you specify in your command, if whatever it is has a flag for the output, like fastqc did, and you give it -o, that will actually be where the location of all of your files that you created will go. They won't go to this out file. So essentially this is the other default. If you don't specify where something goes, everything goes to this. This is what's referred to as standard out. But yeah, you specify that with the command that you run where you want your output to go in a script or in a command below all these submission parameters. Okay. So this is an example of one that I had run. So I was doing a genome generate with star. I gave it a particular name. One thing that you'll have to worry about on this -o and -e, remember you have to give it a different suffix. If I were to say star genome generate for all three of these things, it would be overriding itself and trying to write the same thing in multiple places. So make sure that all of these are unique. By convention, we typically just add the extension .out and .err to the end of these. The .sh you can or cannot do. Particular wall time, and you'll see here, I was reserving 1 node and 8 processors per node, and the queue that I was submitting to is hotel. So these are the different queues that are available on TSCC. You can read more about this on their website. We are going to submit the jobs to hotel. This is a general queue for all users, and think what you will have access to as a UCSD train account. And if you want more information on this, you can find it on the TSCC Quick Start User Guide. Okay, so that's a general overview of how we're going to be operating for the next step, because it's a very memory-intensive job, so we will need to create a submission script and put our command in there rather than actually running it on the command line. Okay, so we're going to be using these submission scripts a lot, and it's kind of annoying to have to write this over and over and over again, PBS, because a lot of this stuff is common. The outline is very common, and the only things that you're going to change are the name of your jobs and what you're reserving it for. So what I recommend doing, I just did this in my home directory, I made a file that I'm calling fake script, and we're going to give it the extension .sh. .sh means that it's a bash script that we're going to be submitting. So you can do this in your home, and then you can add all of these submission parameters. And then every time you want to write a script, I just recommend copying this fake script to a new location with a new name, and then you can just simply add below the specific command that you're going to be using for that given run. It just kind of makes it easier on yourself later. So why don't you guys go ahead and do this in your home, make a file called fake script, and add these general features to it. And remember, you can do it with VI, so you have to use I to go into insert mode, escape to go out of insert mode, and save it with :WQ. Yes, this is all on TSCC. Yeah, this is not on your local machine. And I'm recommending that you just keep this in your home directory because this is something that you will use a lot, and it's a very general use thing. It's not specific to any given project, but once you make copies of this, you will copy the specific script into a given project folder so that when you are looking for scripts that were associated with that given project, it's in a useful location. Show of hands who has a fake script file. Great. Okay. Okay. So next I want to point you guys to the STAR user manual. So the manuals are going to be your best friend for any program that you're going to download and want to use. So it's really important that initially when you look at one, it's probably going to look like it's written in some foreign language and you're going to have no idea what any of it means. That's how I was anyway when I first started. But it's really important that you just become familiar with the syntax of how these things are written because this is going to be your go-to for any questions that you have about how you run these things. So go ahead and open the STAR manual. And it's also really important that the manual that you're looking at matches the version of STAR that you are running. So you'll notice here at the top it says version 2.5.1a. If you go back to your command line and you type STAR -- version, you'll see that that's not exactly the version that we're running, but close enough. So this is a link on GitHub. If instead of using my link, you Google STAR user manual, and you can do version 2.5.2a. And then you click on the first one. Again, this is taking you to the GitHub repository where all the code for this program is stored. And if you click on the manual, it's still 2.5.1a. But it's usually, unless it's like, this third letter changing is very small, minor changes. When it goes to like 3.0 or something, it will have major changes where you might have issues with documentation. Okay, so this is the user manual. Can you all see it? Make it a little bit bigger. It gives you kind of an outline of all of the different things that are available. It teaches you a little bit about how you can install it. So we took a shortcut and installed it with Anaconda because it takes care of all of this for us and it's really nice. But if you were to have to download the code yourself and run the installs yourself, instructions on how you would do that are here. But again, if you're finding a program on your own, I would recommend first Googling if that program can be installed with Anaconda, and if it can, use that. It's simple. Okay. Next, it gives you into a basic workflow. So what you'll read here is that in order to run STAR, you have two general steps. The first step involves generating the genome index files. So STAR requires a genome index in a specific format for each genome that you want to align to. So you'll see that this is the first step that we need to do with the human genome that we're going to be using. And then the second step will actually involve mapping the reads to that genome once you've generated the files. And so it kind of gives you an idea here of which files are going to be required for each step. So here it's telling you that the user is going to supply the genome files that you generated in the first step, so you have to do first step before moving on, as well as the RNA-seq reads, and it tells you what form it wants that stuff in. Okay, so we're going to start with generating a genome index file. So what it's telling us here is that the user has to supply reference genome sequences in FASTA format, as well as annotations in GTF format. And then once you give it those, it's going to generate a specific star index. So again, these are very common files that are used across all species that you're going to be given. If the species has an annotation, it has a FASTA file and a GTF file. A FASTA file is the same thing as a FASTQ, except the quality information is not included, it's just the sequence. So FASTA files essentially give the sequence and associated the chromosome that it's on. It's a very long list of strings of A's, T's, C's, and G's that are associated with each chromosome. And the GTF file, we'll look at those in more detail later, give more information about where the genes are particularly located on all the chromosomes. This is a kind of a more detailed annotation. Okay, so to start, we're going to need to get these files for the human genome HG19. And I'm going to walk you guys through where we get these files. We're going to do a particular example for human HG19, but if you're interested in a different annotation, a different genome build, a different organism, you should be able to follow the same procedure using these very common databases where all of these things are stored. Okay, so if you go back here, I have a link taking you to the UCSC Genome Browser. This is where we're going to get some annotations from. So the UCSC Genome Browser is this nice tool where people have stored a lot of genome information. So we're going to go to downloads and genome data, where this is where the FASTA files are stored. If you scroll through here, you'll see that there's these data sets available for all of these different species. If we are interested in human, so click on human. If you scroll through the human genome, you'll see all of the different genome builds. So every time the genome gets updated, as people get more information, they learn about new genes, they become more and more accurate with where these sequences are actually aligning to, and each different build will start the chromosome at position 0, and then it will have a reference sequence starting there. So those are the things that actually change as the build information changes, because the genes and the locations of these genes are becoming more and more accurate and changing a little bit. So it's really important that you keep track of which particular genome build you are using and keep it consistent throughout. Otherwise, the reference sequence and the chromosome location are not going to match if you're trying to go across different builds. So the most current human genome build is HG38. It was released in December 2013. But the field still hasn't really caught up to it yet. Most things work in HG19, so that's where we're going to go. So we're going to take the build that was released in 2009, which is HG19. And they give you a bunch of different files. So we're interested in the full data set for HG19. And this is what is giving you all of the reference FASTA files. So if you scroll all the way down to the bottom, here are all of the different files that are available. And what we need is a file of all of the chromosomes in the FASTA format. So you can see that's here, it's Chrome FA. So that's telling you that these are all of the FASTA files associated with all of the chromosomes. So this is the file that we need to get the FASTA input for STAR. So we're going to get it in the same way. We're going to copy the link address by right-clicking Copy Link Address. And then we're going to put that on TSCC. So these annotation files are big, so unfortunately we can't keep them in our home, but they are something that you're going to want to keep permanently after you build it. So once we build the STAR index with these files, we can transfer it back to our home later. But for our purposes, we need to start working in Scratch, otherwise we're going to run out of space on our home. So remember, we made a soft link to Scratch from our home, which is really nice, so we can go straight to Scratch. And then within Scratch, I recommend making a directory called Annotations, where we're going to start working with these annotations and put all of the files associated with this annotation. So I already have them. Again, within annotations, you might be working in different genome builds, you might be working with different organisms. So we're going to make a file specific for HG19 and put our stuff in there. So go ahead and make another directory called HG19. And so we want the chromosome FASTA files to go in this directory. So again, we're going to use wget, and we're going to paste the link that we copied. So one thing that I didn't point out when we were using the FASTA files, but that we downloaded from the FASTQ files that we downloaded, is this .gz at the end. So .gz means that the files are zipped. So since FASTQ files tend to be very large, a lot of programs will let you operate on them in the zipped format without having to unzip them first and use all of that space, which is really nice. So we didn't unzip them, we didn't do anything with them. But in this case, we are going to need to unzip it. And this particular file is stored with two different, it's stored as a .tar.gz. So the way that the file extension that you have on a zipped file will tell you how you need to unzip it. So if it's something that's .gz, you can simply unzip it with the command gunzip or gunzip. But because we have .tar.gz, we need to use tar to unzip it. So the syntax to do that is tar-xbf followed by the file name. And when you run that, it's going to unzip the file, and you'll see all of the stuff that's zipped up into this one. So things that it's pushing out, you'll notice that there's a .fa file for each chromosome number in the human genome. There's also a bunch of these other additional random sequences, things that people have found that maybe don't have a specific assigned location yet. Some other exome sequences maybe, or exosome fragments, things like that. So all of this is unzipping, but ultimately all that we care about are these files that end in .fa. So each chromosome number, .fa. So what we're going to do once it's done being unzipped is we are going to remove everything that is not what we want. So if you had a question that was maybe more specific to things that are not, to sequences that are not included in the main genome build, you would be interested in these other files, but for our purposes, we only want the main chromosome files. So what I want you guys to do is use the RM and use the wildcard character star to remove multiple files at a time. So we can use rm-r, remember to do it recursively so that you can do multiple files at once. If you just do rm, it's only going to let you remove one thing at a time. And you can use your star wildcard character to find certain features of files that you don't want. So all of the files that we want have the syntax chr followed by a number, .fa. But if you scroll through here, you'll see there's a lot of other things that we don't want that we need to delete. So can anybody think of how you would use star wildcard to be able to delete all of the unwanted files at once without deleting the files that we want? Basically, you need to identify a feature that's unique about the files that we don't want that does not exist in the files that we do want. So what's unique about the file names that we don't want that is not in the file names that we do want? What?
Random or not?
Random. Okay, so there's a bunch of files that contain the word random that we don't want. So in order to get rid of all of those, we can do rm-r star, and is it capital or lowercase? Random. And we can put a star wildcard on both sides. So what this is saying is remove any files in this directory that contain the word random. And I don't care what's before or after that. So this will allow me to identify all of those files at once. So if I run this, and then ls in my directory, you'll see all of the files that contained the word random were gone. So continue to use that strategy to get rid of all of the files that you don't want until you're only left with the chromosome numbers and the original file that you unzipped. Yeah. Yes. Does anybody know what chromosome M is? What? Yeah. The mitochondria. Yeah. Okay, so these are the sequences that we are going to use for all of the chromosomes. Show of hands, whose directory now looks like this? Great. Okay, so the way that STAR needs to operate is it wants one file for all of the chromosomes. So here we have one file for each chromosome individually, but we need to put them all together into one. So the command to do that is going to be cat. And what we're going to do is we're going to do this for all of them at once. So we're going to do cat chr star, which is going to grab all of the files that start with chr. And we're going to use this caret, which means take the output and assign it to a file. So remember we introduced pipe as the character that will take the output of your first command and put it into a second command. But rather than putting it into a second command, we want to actually assign the output of this to a new file. So we're going to assign this as, what did I call it, all chrome or something? All chrome, so name it something useful and pay attention to the extension of your file. So these are FASTA files, so it's important that you keep them as the .fa and name it something useful. So we're going to put all of these together in one. Yeah?
In the current one, is it also going to work hard?
Yeah, I was thinking about that. I don't think it will because it's zipped, so you can't combine it together with the information. So what is cat doing? Remember, cat's throwing the output of each of these things onto the thing. So what it's going to do is it's going to take the output of the first one and take all of it and put it together into this file. And it's going to do that across all of these things. So when you try to cat on a zipped file, I don't know if that will work. You can try it. You can try cat followed by this name and see what happens.
Is that, so what you have written there, is that going to go to the tar file too?
Yeah, so that's what we were just talking about. So cat is going to open the contents of all of this. Like remember we used cat to be able to look at files. So cat is going to open the contents of all of these things and put it into this into this new file. So it's going to work through each one of these and take all of the contents and put it into the next file. So when it gets to this zipped file, I don't believe it's going to show anything because it's a zipped file. But to check that, we can try cat on there. And then Control C whenever something's dying.
So it's going to keep the compressed files though?
So it will what?
It'll keep the unmerged files though?
Yeah, it will still keep them all individually. We haven't removed them. But it will take the contents of all of them and it will put it into this new file that we need, chromosome.fa. So if you were worried about that as being an issue, how could you modify this cat command to make sure that it doesn't grab that zipped file? So how could I change this using the star wildcard so that it's unique to these files but not this one? Right, so if I added a .fa on the end, so now I'm going to be grabbing all of the files that start with CHR and end with .fa and putting them together. So that will avoid the issue of this other file that's in there. Yep.
Does it matter what order they're in?
No. So for this case, it does not matter what order they're in. Some other programs might require them to be ordered, and you can deal with that accordingly. Cool. Okay. Oh, I already did this. So now we have all of the FASTA files that we need. So that's the first half of the genome build. The second half is that it required a GTF file. So show of hands, is anybody having issues making this chrome.fa file? Good. Okay. So you can see that it exists in your directory of values. Okay. So once we have the FASTA files, I'm going to point you guys to the next, basically, storage bank of annotations, where we commonly get most of our GTF files. And these are found from GenCode. So there's a link to the GenCode website in the drive that I sent out. So the link that I provided actually varies you a little bit into GenCode. You'll notice that I'm already on release 19, so this is chromosome build HG19, and I'm in the human genome. If you just go to the GenCode website, you can navigate through to get into here. And what you'll see here is a bunch of different files that are available for you. So so they also have FASTA files here. This would have been another place where we could get all of the chromosome FASTA files. But we used Genome Browser. You'll notice that these things are stored in multiple locations. So sometimes it matters, sometimes it doesn't. You can read about on this particular website about exactly how these files were generated and what's included and what's not. But so these are the GTF files that we're going to be using. We want to use the comprehensive gene annotation. You guys can read the descriptions to see what these other gene annotations that are available are. But we want to use the comprehensive, and we're only interested in the chromosome. You'll notice it tells you by default this is the main annotation file for most users. There's also this kind of superset of the main annotation file to contain a bunch of other stuff. You guys can read more about that if you want, but the one that we're interested in is the comprehensive gene annotation, the main gene annotation file for most users. And we want it in the GTF format. So again, go over to the link, copy the address, and get that file, and put it in your annotations folder for HG19.
So some people have been asking about what information you publish when you publish. The browser or whatever build you use, whether it's HG19 or HG18 or FM10, that information is commonly seen in your materials and methods. People often say that they got HG19 from CSD or from Jennifer. So that's information you should keep track of whenever you're writing code in the future.
Yeah, and these are things that when you're sharing information with collaborators or comparing data with other people, it's very important that you're speaking in the same language, and in the same language in this case means the same chromosome or genome build, otherwise your files will not match each other. Okay, so once you've gotten that file in your annotations folder, you'll notice that this only has the suffix of .gz instead of .tar.gz. So to unzip this one, rather than using tar, we're just going to use gunzip. And this will unzip any files that just simply end with the extension .gz. And once you've unzipped it, you should see that it's there in your directory. Show of hands, who has a GenCode GTF and an allchrome.fa? Okay. Okay, so now that we have gathered the files that we need in order to be able to run the command, we're going to write a script to run that command. So we'll go back to the documentation in a minute so that we can see what the syntax of that script should look like. But because we're going to be submitting this to the cluster, we need to write a submission script for it. So what I would recommend doing remember we made a projects folder for Lin28B. So I like to have another subdirectory in there called scripts, and this is where I'm going to store all of the scripts that I write that are associated with this project. So we'll specify the output, just like we specified the output in a FastQC folder, an output of star, we'll make a star folder for the output of that. But it's really nice to have all of your scripts in one place, because if you're confused about something that you did a couple months ago and you want to remember how you processed your data set, all of your processing will be stored in one location in the scripts folder. And scripts are generally pretty small, so it's easy to store a bunch of these in your home directory, and these are something that will never get deleted. So again, when you process your data, it's really important that you have your raw data, but also your script for how you generated your processed files, because if for whatever reason your processed files got deleted, you can recreate them very easily by referring to scripts. It's also useful if you're starting a new project and you need to redo a genome build or you need to do an alignment again in a different context. You can refer back to here to remember how you did it previously to be able to apply that to your next project. So within your Lin28 folder, go ahead and make a directory called scripts and copy that fake script that's in your home into that directory. And remember, when you copy it, you want to give it a useful name. So in this case, we're going to be generating the index with star. So the source file that I'm going to be copying is in my home. It's called fakescript.sh. And the destination is going to be in the scripts directory. And I'm going to call it something useful. I'm going to call it star generate index.sh. And remember that when we made this fake script, we just put fillers in for those things that we're going to need to change for each script. So use VI editor to edit this specific script. And you're going to need to change these hashtags. So the queue is going to be the same. We're going to submit to hotel, but give it a meaningful job name. We're going to request, this is a pretty memory-intensive job, so we're going to request 16 processors and 1 node for a wall time of 3 hours. And again, don't forget to give it a suffix specific to the out file and the error file. If you give it the same thing, they're going to override each other. Don't do that.
Are you in GI?
Control C. Control C is like, so what you did is you like used something, but then you finish your command, so yeah. So anytime you're like, show of hands, who has a script that looks like this? Great. So I just want to point out that there are some other submission scripts that we didn't or submission commands that we didn't talk about. But remember, you can read more about the submission parameters. I provided a link here to TSCC. So there's one that you guys might find useful that I've not included on here. You can include a flag to send you an email if there's any error message associated with your job. So I typically don't include email flags because if we're submitting hundreds of jobs, your email inbox is going to get spammed with lots of stuff. But it's nice you can specify if you want an email to know when your job starts, when your job finished, or when your job, if there were any issues and your job had to abort midway through. So you guys can read about that, and if that's something you want to incorporate later on, you can do that. So we're going to go back to the STAR manual. To build our command to generate the genome index. Okay. For each of these are referred to as flags. These are the same types of things that we saw with the bash commands. Whenever we were doing something like with ls, it was -l or -h, or yeah. So the flags that we were looking up at the manual with bash commands. Anytime that the flag is a single letter, it's only going to have one dash. But if the flag is multiple letters or it's something like a word, it's always going to be written with 2 dashes. So anytime you see something that's -- followed by some sort of description, that's a flag that's included as a parameter in your script. So star, the way that we run it, the executable is simply called star. So this is our command. Our command will be star. And then after that, we're going to do each of these flags that will specify how we want this job to run. So all of the flags that are shown here are the different parameters that you need to specify to generate a genome index that you can use for your mapping with star. So I'm going to briefly go over these together with us for the first time, but I want you guys to be able to feel comfortable, so to be able to go to this type of page on your own later to try to figure out which flags you need to include for your submission. So I'll go over the way that you read these and what they mean for this particular example. So first, run thread n. This is the number of threads that you're going to be running on. This is equivalent to the number of processors that you're reserving. So make sure that the number of processors matches this flag that you're going to add with run thread n. And the way that the syntax for these works, you always have a space in between your flags, so it will be star, space, -- flag, space, argument for that flag. So in this particular example, it would be star, space, -- run thread n, space, 16, space, and then you would add your next flag on after that. So again, spaces are very important, and that's convention for how you run flags. The next is going to be your run mode. So you'll notice the difference here. This is written in italics, and this is written in non-italics. So italics is something that the user is going to specify, and something that's written here is going to be, that's what's required. So run mode genome generate, this option directs star to generate the genome. So we will see this change when we go to do the alignment, but this is telling you here for star, the run mode that we're going to use is genome generate. Genome dir, genome directory, this is telling you, and you can see the syntax that we're familiar with looking at, the / of the multiple subdirectories within a file. So this is the full path to the genome directory, and you can read down here, this specifies the full path to the directory, henceforth called genome directory, which is what we'll refer to that as, where the genome indices are stored. So it has to be created with make directory before you run, and it needs writing permissions. So when you create it, it will have my writing permissions. So this is something that you need to make, and I believe it needs to be called star. Or you can, okay, so you can call it whatever you want, but this is where the genome index that you're going to make is going to be stored. So we need to go ahead and make that directory on our TSCC, and then remember the path that we given. So if you go back to TSCC, and we're going to make this in annotations, so go into Scratch, annotations, HG19. So where you have all the other stuff, we're going to make a directory in here, and we're going to call it star. So the path that you would provide for genome dir is what you would copy when you do pwd in that star directory. So we go all the way back to here. So that's the argument that you're going to insert for the flag -- genome dir. And it says here that you need to make it before you run it, so that's why we went ahead and made that file first. The next flag is genome FASTA files. To specify one or more FASTA files with the genome reference sequence. So we combined all of ours into one file, which is really great. And what we will need to do is provide the full path and location of where that file is stored. So when I'm ready to add the argument for that file, what I'm going to do is go to here, and I'm going to ls because it's in this file. I'm going to use pwd, and I'm going to copy this entire thing. And then I'm going to add at the very end, it's the file that's all from.fa. Okay. So again, that's the location in the file that you're going to specify the path to. using the genome FASTA files. And you can kind of see it, they give you like an example. So if you were to keep them all separate, you would have to provide like 23 different paths, which is annoying, but we have one. Same thing with the SGD, or same thing with the GTF file, path to annotations.gtf. This is the annotations file that we downloaded. If we keep scrolling, you can read more about these other arguments. So this overhang, it specifies the length of the genomic sequence around the annotated junctions that it should be used to construct your splice junction database. So we talked about this a little bit. STAR, it works very quickly. It deals with spliced reads because it uses information known in these annotations about where these splicing junctions are. So let's see. It kind of gives you some hints. It says ideally this length should be equal to the read length minus 1. So if you have a paired end, 100 base pairs on each, then the ideal length is 99. In cases of varying length, you should use the max read length minus 1. In most cases, the default of 100 will work as well for the ideal value. So in this case, we can just take the default of 100. Okay, so that will be enough information for us to go ahead and run this. If you want more detailed information, you can read about it in the advanced options. It will tell you about where you find FASTA files, where you find GTF files, so all of the work that we've already gone through together, as well as a couple other useful things. But for our purposes, these commands will be enough. So we need to add this to our submission script. So I'm going to go back into the directory where I have the script. This is in projects. Scripts. And I'm going to open the script that I made called star genome generate. Okay. And then one useful hint for VI editing, if you're not in insert mode yet and you want to jump to the end of the line, you can use $, will jump you to the end of the line, but that won't work if you're in insert mode. So I did that before I went into insert mode, and then do I, just get to the end of the line quickly. So below your submission parameters is where you're going to add this command. So the command is going to be star, and then I'm going to start adding my arguments. So first it's going to be run thread n. I'm trying to put this so you can see both of them on the screen. So space dash dash run thread n, and the number of threads that I'm going to give it matches my processors at 16. Next I'm going to give it run mode, and the mode is going to be genome generate. Next I'm going to give it the genome directory. So when you get to genome directory, this is an important thing where, so tabs won't work in a VI editor, but you want to copy and paste to avoid making errors. So what I'm going to do is, as soon as I get to the genome directory part, I'm going to save and quit this. And I'm going to look on my terminal for where I know this genome directory is. So I can just simply use ls. I know it's in Scratch, Annotations, HG19, and star. Okay, so I was able to get through all of that with tabs, so I know that there's no errors in here. So I'm going to copy this entire thing, go back into editing my script, and I'm going to jump to the end of the file, and I'm going to add that with paste so that I don't make any errors. And I'm going to keep going with the other arguments. make sure you guys pay very close attention to spaces. Typos are issues. Make sure you don't have any typos in your argument names. Capitalizations are important. And avoid typos in your packs by copying them all first. have a submission script that looks like this? Raise your hand if you do not have a submission script that looks like this. Okay, lots of people just didn't raise hands. So we're still working on it a little bit. So once you have a submission script that you're happy with, you're going to submit it to the cluster. You guys, I've included this on the file that we're working from. So the command is down here. So you can refer to that if you want to while I take this away. But for those of you that are done, I want you guys to go ahead and submit this to the cluster. So we're in the directory where the script is located. So I can just call it directly. If I wanted to call it from my home, I would have to give it the full path. To submit to the cluster, you use qsub followed by the file name, and then press enter. So qsub submits it, and all of the submission parameters that you've specified are in the .sh script. So it's going to create a job for you, and you'll see it's a job ID number. This is for those of you that are still working on this.
So could you do this just to use QSAC to submit any script that's running in the program?
Yes, so qsub always submits a script. Your script is what you change based on the command that you want to run or the parameters that you want to specify. So qsub is essentially just sends all that information remotely to the cluster.
And then SH is the script of this.
Yes.
And then I were like down in my script to specify that it start, is that I can start at the beginning.
Correct.
And then.
So that's just a start command. That's what this is. So this is the command that I want to run. So if I were to be running, yeah. So this is the executable. So this is just some general syntax about things. This star chunk that we've run is the command that we're running. If we were to be doing this on the head node on the command line, you would have to write this full thing. So the star component is the executable. It's the same thing like if you were to run ls. It's the particular command for that program. It's the executable. And each of these options are your flags. So all of the dash whatever are all of your different flags that specify for this program. So if we had enough memory to be able to run this on the head node, and we just wanted to type this in one line on our command line like we have been doing for other things, we would have to type this entire thing. So all of this is one command running on the star executable with these various flags describing the way that we want star to run. And it's submitted to TSCC with the specific job parameters that we've specified at the top. So we're going to use this format over and over again with different commands or different submission parameters depending on what our goal is. Raise your hand if you've submitted this script. Great. Once you've submitted a script, you can check the status of your script with qstat, followed by your username. So whenever you do qstat, it's going to list all of the jobs that you have submitted to the cluster. Your job ID is this number that's right here. It's going to tell you the username that submitted it. Mine's Train 01. I don't know why the one's missing. The queue that you submitted it to, we submitted it to hotel. The name of your job. It tells you the nodes and processors that you requested. It shows you the wall time that you requested. And it tells you the status. So the status, you're going to see three different letters here. Q means that it's sitting in the queue. It has not started yet. We're waiting until the available resources are there. R means that it's running, and C means that it's complete. So if it's running, your output files will not be there yet. C means it's complete, and you should be able to find, if you had in your script specified an output, that output should be there. You should be able to look at your .out and your .err file to read more about how your job actually ran. Is anybody's running right now, or does everybody say Q? This is a very high memory intensive job, so it might be Q for a while, but that's okay. We have some other stuff that we can work on in the meantime. Does anybody need help getting their script to the point where they can submit it? Okay. I want to introduce something else that's really useful, and we're going to get back to using our bash RC. Oh, Julia, if you want to look at her thing, she's getting a weird error, and I can't explain it. Okay. So I'm going to introduce the concept of an alias, and an alias is something that we are going to define in our bash RC, and it's going to be part of our environment now. So all an alias is it is renaming a command and something that you're going to be using a lot. You can essentially give it a new name that's shorter and easier to use. So for example, we just looked at the submission of our job with this command, qstat-u space followed by your username. It's really annoying to have to type this out the entire time. It's kind of a long command, and you use it a lot. Basically, every time you submit a job, you're going to be checking the status of it with this command. So what I'm going to do is I'm going to replace the syntax of this command and permanently store it with a new command name, or with a new syntax. So I want it to instead be qme, and essentially qme is going to replace this command. So whenever I run qme, it will be like running this command. And the way that I do that is I define that as an alias in my bash RC. So if you open up your bash RC, at the bottom, you can add, and you can do this for whatever you want. You can try this out for yourself later if you find yourself running a lot of these commands very regularly that's annoying to you and you want a shortcut. You can add a specific alias. So in this case, we're going to define it as qme, which is what we want to replace this entire thing. So now on my command line, whenever I run qme, it's going to think that I'm running qstat-u followed by my username. It's just a little trick to add my own keyboard shortcut. Some people will do this, like for example, I know Gene likes to use ls-ltr a lot. That's how he just like by default likes to look at the files in his directory. So he's made an alias that says ls-ltr here, and he's redefined it as ll. So essentially giving it a new name that's shorter and more convenient to run. So why don't you guys try adding this line to your bash RC, and then go out of your bash RC and try using it. Yep, good call. So did you try using it? Or you, okay, did anybody try using it and get an error? And what does your error say? Command not found. So why didn't it find the command? Yeah. So you changed it and you have to update your changes with source ~/bashrc and then try it again. Is anybody still not able to get QME working after sourcing your bashrc? Yes. You did? There's a mistake in the documentation that I've provided you guys, which is why some of you are probably getting this error. I accidentally put a space in between the dash and the U on the folder that I gave you. Make sure that there's no space there. You guys should know that's a problem because anytime you use flags, and -u is a flag for the command qstat, it always has the syntax of dash followed by the flag character, space the argument. So you see that there's a dash but a space in between the U. That should automatically be red flagged to you guys that that's going to be a problem.
Are there any other shorthands that you recommend that we technically have?
Yes, but we haven't gotten to it yet. So one that you can do, actually, this would be a useful shorthand. If you go to your home computer, and I'm not sure how this will work on the Linux systems, but you guys can check. If you go into, or if you list the files in your home directory with -A, you're going to see all of these things. This is essentially, this is on your computer, not on TSCC. So for Macs, the bashrc equivalent is the bash profile. So you're going to open up that file. I'm not sure what it is on Linux. You guys will have to check and see if you can figure it out. But if you open up that bash profile,
What does it mean if I don't have that?
that's what I'm not sure. Hang on a second. So if you run vi ~/ and use tabs to complete your bash _ profile, you should have one of those. Okay.
Only have history and session now.
Okay, I'll look at yours in a second. But here's another alias that I've added in my bash profile, and I've called it TSCC. So now when I want to log into TSCC, rather than typing all of this, I can just type TSCC. And this is, again, this is on your local machine, not on TSCC. went to home to see. So I'm not sure, does anybody else not have a bash profile? Okay, I'm not sure exactly how I created this, so I can give you guys more information tomorrow when I look into it. But this is another idea that you can do aliases both on your home computer and on spaces. And I don't remember where my bash profile came from, so I'll have to go back and look at that, and I can share it all with you. Okay. Go back to your stats.
Oh, no.
Oh, okay. Good. Okay, so one other thing that you guys should be able to use with these job submissions. Okay, so mine is still sitting in the queue. Others, if you realize after you submitted it that you made a mistake, but it hasn't finished running yet, and you don't want to wait until it's finished to kill it so that you can fix your changes and then submit it again, you need to delete your job. If you try to submit your job again, but without deleting the first one, they're going to conflict with each other and start overwriting files and be very confused. So if you ever need to delete a job, the syntax to do that is qdel, followed by the job names, or the job ID. So you would copy this number and run that, and that's how you would delete your job. And then after you delete it, you can check your status with qme, and you'll notice that this will go to a C as completed because it's been deleted. Raise your hand if you're confused about the stars script that we submitted, or if you're confused about how it's working when we're submitting things to the cluster. Anybody confused? Okay, cool. So since those things are going to sit in the queue, they may take a little bit of time. So tomorrow we're going to look at the output of that. But in the meantime, I want to go ahead and get the alignment started. And in order to do that, we're going to use a star genome that I've already built for you that you can use as your directory for where the star genome is located. Once yours finishes running, you'll be able to input your specific directory in this script instead. So that is located in /projects /ps YOLab. And keep in mind, this is a different branch of the root node, so this is not your home, so you're not using utility here. We're going into the other branch known as projects /ps YOLab mstp _bootcamp _2016, I think, /star. And I'll double check that path is right. So you guys should all have access to this. If you try to find that file or that folder, you can list what's in it. And it should look like that. So this is the full path, and you guys can use tabs to navigate through it. And this is the content of that file. Is anybody not able to see the content of that file with LS? Okay. So we're gonna look at the submission parameters that we're going to use for the alignment, and this is the location of the genome index that we're going to use as opposed to the one that you just made. And again, once your genome index finishes, and if it finishes correctly, you'll be able to import your folder in this location instead. Okay. So for this, we're going to make another script. So we're going to go on to step 2 in star, which is mapping the reads to the genome. And we're going to write a new submission script. So again, copy the fake script that you made and give it a new name. And I'm going to call it star mapping.
Can we do this even though we're in star and we need to get out of it?
What?
Do we need to go back to our own portal? Like, do we need to get out of the Now lab first?
Oh, yes. So, well, when you copy, you're going into this home directory. So again, if you were to try to copy this from whatever location you were in, as long as you run it like this, it should work. If you're trying to copy it without giving the full path of the file, then you first need to be in the directory where this is located. So why don't you guys go ahead and make the changes to that file on the submission parameters. I don't remember. So for this job, we're going to use nodes equals 1. We're going to try ppn equals 8. And we're going to give it a wall time of, it won't take this long, star is super fast. So we're going to give it just an hour. And then make sure that you give it a useful output and error file. Okay, so I'm going to encourage you to not cheat and look at the command that I probably already put in this core document. Take the next 5 minutes to work with the people around you, discuss these options, go to the STAR manual, go to the different submission parameters for running mapping jobs, read the different arguments, and see if as a group among the 2 or 3 of you next to each other, you guys can understand what arguments you're going to put for each of these flags and which flags you want to use. So you'll notice here, this is the same thing that we went through with the genome generate. All of the flags are here with descriptions. These are the kind of basic ones at the top. If we scroll down, it tells you a little bit more about advanced options. One thing that's really nice about this manual is it gives you the encode options. So this is kind of like an example of field standards that are currently being used for these mapping jobs. So if you're not sure what value you should use for any of these flags, here they give you an example of kind of what a typical alignment job would look like. And you guys can read about those. So you don't you can write them down on a piece of paper, you can write them in a text file that you want now, but work with the people around you to essentially build your STAR command for mapping. And why don't you guys spend the next 5 minutes doing that, and then we'll go over the final flags that we decided on as a class together to make the final script. And like I said, avoid cheating by looking at that document. I mean, you can if you want, but what fun is that? And make sure you talk to the people around you, help each other out. Are you guys helping each other and talking to each other? It's kind of quiet. Yeah, so I'll see if they can better command. They can, and they can submit it, and I'll have them do that. For the rest of the files, it's okay.
Is to transfer the files.
Oh, okay. No, just use this path. Okay, so let's reconvene. There's only 5 minutes left, so I want to talk about what you guys learned from these parameters, and we'll go over them all together. So, what are you going to put for run thread n? Are you going to use, first of all, are you going to use it? Yes or no?
Yeah.
And what's your value going to be? Okay, 8, because we're matching the number of processors. Good. What are you going to use as the genome directory? Great, it's this path. So you don't need to actually copy that directory into your whatever. You can just provide the full path of it as the argument here. See how it says path to genome directory? So this is the path that you're going to provide. And then again, once your genome is finished, you can swap that out with the path that you made. Okay, read files in. What is this? Somebody raise their hand to explain this slide for me. Any ideas? Yeah, Andrea. Your which files?
Your files that you downloaded earlier.
Great. So, okay, yeah, it tells you right here. So these are the files that we downloaded. All right. Did you guys learn about the paired end? So we have 2 reads. So we need to specify read 1 and read 2. And it tells you that if you're using paired ends, you need to have both of them. So you see how here they have this bracket? That means that the second argument is optional. That does not mean that you need to include the bracket in your command. So what it's going to look like is -- read files in space path to read 1 space path to read 2. And the read 1 is going to end with .fastq.gz, which is the read 1 file, and the read 2. And you might have to double check back on the ENCODE website with the specific read ID. I know those are not really named very easily to be able to tell which one is read 1 and read 2, but you can check from your download. Okay. Oh, that's the same thing as up there. Okay. Did you guys learn anything else about read files in that you're going to have to include? Okay. So the other thing you need, it tells you in here about if files are compressed. You're going to have to specify that in your flags so that it knows how to read it properly. So because we're using files that are compressed, they have that .gz extension. It tells you right here. For example, gzip files use the -- read files command followed by zcat. Or you can use this. And then this is for a different type of compression. You're going to use this. Okay, so we're going to need to include the command read files with zcat. So I want to go through a couple of the other ENCODE options, which are things that we're going to include, and these are decisions that you as a user can make based on your particular experiment. So we're going to filter out by SJOut. What this is telling it is to use the splice junction database that we're providing in the annotations as a way to help increase the confidence in the splice junctions that we're calling. So star will very nicely use the information in the annotation to automatically look for specific splice junctions, but if it's also finding information supporting splice junctions that are not in the annotation, so these may be novel events, it will still pick those up, but it will require more reads to support that, and it will require a longer overhang on each side to be really confident that that's actually a splice junction and you're getting mapping on both of those sides. But if it's something that's well annotated and well known, we don't need to have those safeguards, so it's maybe only going to require one nucleotide overhang on each side. This is another really important command. It talks about multiple alignments. So a read with a given sequence can map to multiple locations in the genome. This is particularly important for repetitive elements. So if you have a repetitive element that your read is mapping to, it might map to multiple spots across the genome, and it will be really hard to tell exactly where that was coming from. So some people like to be as strict as possible and say, I'm only going to accept the read if it only maps uniquely to one location. Most people will set this to a parameter of 10, so the ENCODE is a little bit more lenient to 20, and we'll be able to see on the output file how many of our reads are actually mapping to more location, how many mapping things are unique, and that's something we can make a decision on downstream to decide if we're going to include reads that map to multiple locations, in which case we might be double counting, or if we only want to use unique reads, but that's what this flag means. This overhang is what I was referring to before. So for unannotated junctions, you want to have a longer overhang to be really confident that what you're seeing is actually a splice junction. But for annotated junctions, one nucleotide overhang is fine because we know that that exists there. It's something that we're looking for. And if that one nucleotide on the other side matches, we're pretty confident that that really is a splice junction. Again, these are things that you can play with. Outfilter mismatch. So this is the maximum number of mismatches per pair of reads. If you just give this a massive number, it switches off this filter. So if you want to be very strict and not allow any mismatches across your read, you can reduce this number to, say, 0 mismatches across the read. I'm only accepting perfect alignments. We realize sometimes there's SNPs relative to the genome. There's often a lot of variations, so you don't typically want to do that. And you can just switch that filter off with a max of 999. So they give you an example of a typical filter that you might use. So for example, I guess a 6% mismatch rate is considered standard. So if you have paired end reads, then you would have a value of 8 here. The minimum intron length that we're going to allow is 20 bases, and the maximum intron length that we're going to allow is 1,000,000 bases. And the maximum genomic distance that we're going to allow between databases is also 1,000,000. So these are the parameters that we're going to use. Sorry if you have more questions on these, we can work on them more next time. I just wanted to get through this really quickly before we ran out of time. And so what I want you guys to do for homework is to make another .sh script, which we just copied, and we have the parameters specified. And I want you guys to add the command for star using all of those parameters that we just talked about. So I've included it here in the notebook for you. I guess the one that we didn't talk about, out file name prefix, this is where you're going to specify where the output of these are going. So we're going to be making alignment files, we're going to be making BAM files. We need to specify the output of where this is going to go. So within my Lin28D, sorry I missed the B here, projects folder, I'm going to make a new directory called star alignment. And each alignment is going to generate multiple files. So it's asking you for a name prefix. And that prefix that I'm going to give it, since we downloaded Lin28 knockdown data, the replicate 1, this is the prefix that I want all of my files to have. And then every file that gets generated will have its own suffix depending on what the file is. But I can match them to my experiment because they will all start with this. Okay. Are there any questions about this command before I set you free to do it all by yourself as homework? Yeah?
So what is a genome load?
Genome load and remove, it's a memory issue thing, so essentially it's rather than loading the genome and storing it in memory, it's going to load the annotation and then remove it from memory when it's done with it. It's a way that makes it process faster. And you should be, if you have any more questions about these, you can go to the manual and find that particular flag and read more about it. So we're going to start class tomorrow. Chances are you guys will probably get some error messages. Your jobs might fail. You might have typed something in wrong. You might be using something incorrectly. You'll see those in your .out and your .err files associated with your script. So pay attention to how your stuff runs. Feel free to take a look through your error files, your output files. We're going to start class tomorrow by looking at example errors and outputs errors and out files that you guys have stumbled across. And if anybody has good results with files that we've made, we'll take a look at the output of those as well. So that's what I want you guys to do for homework. Write this submission script for the data set that we've downloaded and submit it as a job. Yeah?
When you're writing the class Q files or command lines, can you specify a wildcard just all the class Q files in this directory?
No. So for star, we need to specify that this is the FASTQ file, this is paired 1, and this is read 2. So I don't think that star will let you use the wildcard because it needs to have it in a very specific format for this particular alignment that you're doing. It wants read 1 to come first, and it wants read 2 to come second. Yeah?
Can we connect to TSCC from anywhere, or do we have to get UCC?
You can connect from anywhere, except for the Linux users will not be able to, because your login is specific to the class. So for you guys, you can use a text editor to make this script, and then you can submit it when you get to class tomorrow. Or if you send it to me in an email, I might be able to submit it for you. I can definitely submit it for you. So if you just use a text editor on your computer, like TextEdit, to have all of this, I can submit your job for you. Or if you would like, you can just submit it when you get to class tomorrow, and we can look at the outputs and you'll be able to see what other people are getting. Also, this lab is open from 7 a.m. until 7 p.m. So if you want to come back and work in this lab or any other, actually I don't know how it will work outside of this room. So yeah, I understand with the Linux users you'll have a little bit of a harder time doing this, but we should have enough outputs for people that are doing this on their own computers to be able to work on this together. Yeah?
Log out or take
Yes, so once you submit your job, it goes away to TSCC, you've remotely pushed it, you can log out and your job will still be running. And remember, you log out and exit. Okay, sorry for going a few minutes over today. And also, there's no lunch for you guys today, but we'll do lunch again on Friday. And yeah, so that's your homework for tomorrow. Any more questions? Okay, thanks guys.